First of all, a very warm welcome to everybody here. Thank you for coming along to RCSI's MyHealth lecture, which this evening is on epilepsy. I'm just going to take a couple of slides to give some very broad background to epilepsy, and in particular a little bit about normal brain function versus how the brain may be slightly differently working in epilepsy. After that, I shall give a short presentation which will highlight some of the research areas that my own laboratory here at the College of Surgeons is interested in, but also speculate a little bit about where treatment and diagnosis of epilepsy is going into the future. So before we get started, let's start with the easiest question of all, and that is, what is epilepsy? My guess is that members of the audience here may have family members with epilepsy, they may have epilepsy themselves. But the audience is also probably made up of people who've been attending some of the MyHealth lecture series and are interested in learning a little bit more. So I'm going to begin with some basic introductions. So first of all, epilepsy is a brain disease. It's often also referred to as a neurological disorder, but it is widely referred to now as a brain disease. What is the key clinical characteristic of epilepsy? It is seizures. So if you have epilepsy, you have a tendency to having seizures, and more than one seizure. So what is a seizure? A seizure is caused by excessive activity of the excitable cells within the brain. These excitable cells are called neurons. So epilepsy is a disease characterized by recurring seizures. So to understand a little bit about what a seizure is, we're going to first have a look at how, in a very simplistic way, how the normal brain works. Our brain is made of about 100 billion excitable cells, which we call neurons. And these neurons are very specialized to send little electrical signals to one another. So the way the brain works is essentially sending these little electrical signals. And they send them down these long projections, which we call an axon. When that little electrical signal reaches the end of the axon, it releases a little puff of a chemical that we call a neurotransmitter. That neurotransmitter can do one of two things. It can either excite the next cell, or it can inhibit the next cell. And normal brain function is made, occurs as a result of a balance between this excitable activity and then these other cells that work to dampen down activity within the brain. So under normal conditions, individual neurons fire these electrical signals as they send and receive information. What happens in a seizure is that groups of neurons within the brain all fire at the same time. They synchronize. And large numbers of these neurons can fire at the same time. What I'm going to play next is a short audio clip. So in this clip, and you can find lots of this fantastic stuff on YouTube and these other sites that show how the brain works normally and also gives you an idea about what happens during a seizure. So what you're listening to is, and it's quite an old-fashioned way of recording it, is electrical impulses in brain cells. And it's recorded with a device called an oscilloscope, and these have been around for a very, very long time. And what you're going to see on the screen are little electrical pulses from individual neurons. And they've sort of connected it up so that you can actually hear when these neurons are firing. So those little spikes, that's when the neuron fires. Okay, yeah. So that's a sort of a normal response of a neuron. That neuron may have been responding to signals that it was receiving, and they can fire quite rapidly, so hundreds of times a second. But during a seizure, what's going to happen is all of these neurons are going to fire at the same time. And that's very disruptive to how the brain likes to work. So if a seizure occurs, it's going to basically interfere with the part of the brain that the activity that part of the brain does. So if that seizure occurs in the part of the brain that controls your body, then you can get these convulsions, which is one of the things that people are very familiar with when they think of a seizure. But that activity may occur in the part of the brain which is involved in processing sensory information, like things you hear and see or smell. And so you can get quite different experiences as well. There are two main types of seizure we talk about. A focal seizure. This is when the seizure activity begins in one part of the brain. It may stay there, or it may spread into other areas of the brain. And then we have a generalized seizure. So this is when that seizure activity occurs on both sides of the brain at the same time. Essentially what we're talking about is a brief imbalance within the brain between excitation and inhibition. And almost any sort of injury to the brain can cause that. And this is injury in a very broad sense. So this could be something to do with something that happened early in your life. It could be a trauma to the brain. And it could be a genetic factor as well. And we'll learn more about some of the genes that have been identified that, if mutated or altered, affect how neurons fire. And as a result of that can lead you towards having epilepsy. So that finishes the short introduction. So we come now to a short presentation by myself, and it's fairly humbling to follow two clinicians who really changed the lives of patients with epilepsy and their families. And I've worked on trying to understand what causes epilepsy for 20 years, and I probably haven't helped anybody yet. But I'm optimistic that we will change that in the future. So the idea of my talk is to cover a little bit of the research that we've done at the College of Surgeons here, and then kind of move into a couple of other areas and sort of give a slightly optimistic, futuristic view of where we might be in a few years. Some of these things will happen, and some of them won't happen. And, it's difficult to know which. So look, I'm going to touch on three things. The first of them is new types of drug. So we have about 20 drugs for epilepsy, and they work, as we know, in about 2/3 of patients. But even the patients that they stop the seizures in, they don't seem to treat the underlying cause of the seizure. So if you take the patient off the drug, the seizure comes back again. So the epilepsy is still there for many patients. Future treatments should be disease modifying. We've got to get drugs that actually affect the disease, prevent it, stop it, and make you seizure-free. The second change is around genetics, and we've heard a lot about this already. I'm going to touch on some of this. This is going to be transformational across all forms of medicine, but particularly for epilepsy. And then the final area, which is something quite outside my sphere of ability, is around monitoring. And so I'm going to touch on this very briefly at the end. So first of all, treatments of the future. When I started research, the thing I was most interested in was kind of understanding what happens to the brain when you injure it. And what we've learned, and what many others working in this field have learned, is many different things happen that are all contributing a little bit to creating a brain that triggers seizures. And what we've learned is that if you just take one of those things away, it's not enough to stop seizures. So a drug of the future, a disease-modifying drug, probably has to work on several processes at the same time. Because we know that within one of these regions that currently Dr. O'Brien is removing, within that region we have damaged neurons, we have dead neurons, we have inflammation, we have new neurons being made, we have neurons wiring up differently. So how are we going to tackle all of those different things? So one of the things in my lab is to think about sort of controllers of multiple genes. Is there something that the cell makes that could have an effect on an entire network of genes? And about 10 years ago, a new molecule was discovered that is present in all cells in the body, but particularly within the brain. It's a type of nucleic acid, so it's a cousin of DNA. And this molecule, it's called a microRNA, is produced within cells, and its basic function is to stabilize and control networks of genes. So it has small effects on many different genes, generally dampening down their activity. So our idea was this: maybe there's one or two of these microRNAs that are made inside brain cells that have effects on just the right kind of pathways. And if we can target them, maybe we can then disrupt the epileptic brain in a way that we can't do with current drugs. So this was something that we tried to do. We started off, we collaborated with Norman and with Donica, and we had brain tissue that was donated to us by patients who had gone for surgery. And we also have experimental models of epilepsy where we try and kind of recreate what's happening in a human in a lab. And we analyzed all of these different microRNAs, and we identified one that really caught our attention, because it was known to be involved in sort of the structure of neurons and how they communicate. But the problem was that there's no simple way to target this type of molecule. Its structure was a little bit like DNA, and there was no traditional drug-like molecule. So the first challenge for us was to try and block how this molecule was working. And so we actually had to kind of create a little sequence that looked a bit like DNA that would glue on to our molecule. And so we designed that molecule, and then what we found, which was remarkable, was that it seems to block the function of this for a very long period of time. So anybody who's familiar with treatments for epilepsy, you often have to take a pill 2 or 3 times a day. So this one treatment would last for a month. So that was something that was very exciting for us. And so we tested this molecule in I think 7 different models of epilepsy. So this is not in humans, this is in models of epilepsy, and it worked in 6 out of 7 of them. So we went through all of these, and the most important of all was that in at least one of the models, it didn't just reduce seizures, it just stopped them happening again. So when we sort of studied seizures that were occurring in a model of epilepsy, they essentially stopped. So we were very excited about this, and the next challenge is, of course, how do you test a molecule like this? Because this type of molecule's never gone into humans before. Until a couple of years ago, when a pioneering Danish company showed that you actually could block this type of molecule. It was for a liver cancer at the time, but they showed that in principle these molecules could be tolerated by people. And there's now this group of companies, a lot of them in America, that are starting to try and target complicated diseases by going after these molecules. And we started talking to one of them about how we would like to develop this new type of treatment for epilepsy. So we've just begun this work. This is only we're about 6 months into the project, and we're testing their version of this molecule in models of epilepsy. So this is very exciting for us. Okay, so the second sort of future topic is around genetics and genomic medicine. So as Dr. Delanty has said, the cost of sequencing your whole genetic code is is becoming lower and lower every year. And hopefully there will be a time in the near future where that's one of the first things that happens if you present with a seizure. That we can look at the genetic code, we can see if there's an error somewhere. And there were some really fantastic examples and work done here at the College of Surgeons. So one of the studies that was published by Norman and his colleagues was about a very rare reaction to a common epilepsy drug called carbamazepine. So in very rare cases, patients would have a horrible skin reaction to this drug. And they discovered that there was a particular genetic mark that predicted who would have these reactions. And so now, every bottle, every safety instructions for this drug now has this warning about this genetic issue. So it can be very, very powerful. And then there's one or two cases where a child has been found to have a genetic mutation, and it turns out that a drug used for something quite different will actually work on that. And we're seeing sort of every couple of months some new type of drug coming out, which is based and tailored towards that patient's own genetic code. And then the other really transformative technology, which I think is going to be used more and more in the future, is that we now can grow skin cells from a patient, turn them into a neuron, and then study how that neuron behaves. And actually, we can wash over all sorts of different drugs and see if there's one particular drug that stops that seizure behaving, that brain cell behaving abnormally. So this is very, very exciting times. Another unique first for Ireland is the development of an electronic health record for patients with epilepsy. This was the first national electronic health record in the world specifically for epilepsy. And it was done in Ireland, and it was done by researchers at Beaumont Hospital in Norman Delanty's team. Why is this important? It doesn't sound very exciting, right? Well, it can open up all sorts of opportunities. So it can help with recruiting patients to clinical trials. Every specialist centre in the country is all linked together. It's a way that if you're carrying a monitoring device, that you can upload your data and your doctor can see it without having to wait weeks for an appointment. So this type of development, and it's being led now by something called eHealth Ireland, this is going to transform care in the future. And on that subject of monitoring, there's great advances going on now in terms of things like epilepsy watches, so sensor devices, a little bit like Fitbits, which respond to seizures and can feed back information both to yourself and also to your doctor. We've heard about brain stimulation devices. This is that vagal nerve stimulator. But I want to tell you about something called optogenetics, which is even further into the future, but very, very exciting. So a number of years ago, scientists discovered some light-activated proteins. They were found in very ancient sort of bacteria-like organisms. And they turn on and off in response to light. And what they do is they either activate or inactivate a cell. So some scientists put these genes from bacteria into brain cells. Now, you can turn on a brain cell or turn off a brain cell by shining light on it. And this is shown in most of this work is, of course, done in animals at the moment, but this particular rat has been injected with one of these light-sensing genes, and its behavior, and it could be seizures or it could be something else, can now be controlled by turning light on and light off, because then brain cells will turn on and off. And there's a lot of interest in trying this in drug-resistant epilepsy, so that rather than having an electrical stimulation here, you'd insert one of these light-sensitive channels, and then any time there was a beginning to be electrical activity, it would turn on the light, and the excitable zone in the brain would shut down, and then it could turn off and you'd carry on. So these are sort of potential future approaches in epilepsy. And then one of the other areas that we're particularly interested in, and again working with Norman in, is around biomarkers. So we've heard already the difficulty of diagnosing epilepsy, and it would be wonderful if there were a molecule just in the blood or some other fluid that gave you an idea that that patient had epilepsy. And this links back to what I said at the start about these new molecules called microRNA. We've actually discovered that patients with epilepsy have higher levels of this molecule in their blood compared to healthy controls. So there's a long way to go, but we're optimistic that maybe some combination of these molecules and other diagnostic testing would one day lead to support for epilepsy diagnosis to speed it up. And at the moment, researchers at RCSI and hospitals and Trinity College and other centres are sort of trying to come together and bring these three ideas together at the same time. So future treatments, future diagnostics, and then this wonderful electronic health system that we now are beginning to roll out in Ireland. So thank you for your attention. Hope you've enjoyed this little short journey into the future. Hopefully it's not all futuristic and we will get some of these things into practice.